Okay, let's begin. We've got slightly more material than usual today. However, possibly on the upside, this Thursday we will not be having lecture. Sadly, I will have to be, I'm giving a talk in Atlanta, and so I will not be here on Thursday. I'm sorry. I actually like your response. Usually you tell a class you're not having lecture and everyone cheers, and it really bums me out then because it means they don't like it. Anyway, so we're going to be able to get everything covered, though. I've been sort of packing stuff in so that we don't have to leave anything out. Now, today we're going to cover my own personal opinion. I think some of the most interesting things we're going to talk about this entire term in terms of what goes on at the synapse. So let me first recap what we covered last time. We talked about neurons and how they work, and we said that first you had a signal that was initiated somewhere else in the body. So this was your fingertips detected something, or there was a chemical on your tongue, or you smelled something and it stimulated these dendrites that were the sensory receptors. They're going to be somewhere in your body, and as soon as they were stimulated, they started a signal. They started the action potential that was traveling from the dendrite through the cell body. Then it goes down to the axon. We looked at what happens in the axon, right? That there was, you shouldn't even really have to write this down because we talked about this last time. You had that electrical signal. The signal causes the sodium molecules to rush in, making it radically more positive, electrically charged. And then you had the other gates that open up and the potassium rushes out. That restores it back to its negative charge. Then we had the little pump that pumps the sodium back outside, the potassium back in, and you reestablished it. This moves all the way down the axon until it gets to the synapse. And at the synapse, we also use it as a verb. We say it synapses with either a muscle, where it hits the muscle and it causes a contraction to occur, or it bumps up against another neuron, and it can either cause it to fire or it can cause it to not fire. You have some that inhibit firing. So these were what the cell bodies looked like. They had all these branching dendrites that were receiving signals from the outside world and from other neurons. And then you had the long axon. Here's another one. You can stain them in a lot of different ways. But there's just these large meshes of connections between them so that they can cause signals to move from one to the next. Here we have lots of axons. So you have a bunch of cell bodies up here, and then their axons are reaching out really far here. They might be going all the way to the spinal cord. Here's where they make their connection with muscle cells. And so it's just like wires that are clamped in. So your brain sends the signal down. It moves down the axon. It gets to the muscle cell. It lets out neurotransmitters, which we'll talk about today. And those cause the muscle to contract. Here we have a bunch of neurons that are making connections in the spinal cord. But same thing. You see that the axon ends, and then you have a connection with this other tissue. So what we'll do now is look at what goes on at the synapse, because what goes on here is going to be related to sensations that you have if you take medicinal drugs or recreational drugs. I've got maybe the worst overhead of the quarter necks, but it's not as bad as it looks. Or maybe it is. I'm going to leave this up here for a while, though. Here's the general picture of it. You've got the axon, and then you have what looks like a shower head. And the shower head has a bunch of these orange things filled with chemicals. Just don't write it down for a second. I'll give you time. These things, when the signal comes down, it's going to cause these things to bubble to the front. They're going to fuse their membrane. They're going to dump the neurotransmitter out. It's going to float across this little space, land on a receptor, cause these channels to open up, which is going to cause sodium to rush in, and then you're going to have this either muscle cell contract, or if it's another cell, it's going to fire. Then The neurotransmitter is going to fall off. It's going to float around until it gets taken back up, repackaged, and the whole thing starts over. All right, I'll give you a minute to write that down. I wouldn't have you write this down if it wasn't really important. We're going to see that almost all drug effects that you will ever have any experience with are a function of modifying what goes on right here at this at the synapse. And as you can see from the actual photo here, the diagram captures pretty closely what it does look like in real life. And then what we'll do is we'll go through each step, because various drugs that you may be familiar with all modify what goes on here in slightly different ways. ACH here stands for acetylcholine. That's the neurotransmitter that's inside the orange vesicles. The myelin sheath there is, remember, just the fatty substance that insulates the axon so the signal can move more quickly. It looks like just another sort of chemical set of structures inside the body, but here is the place where the chemicals can actually influence your behavior. So an increase or a decrease in a tiny little chemical actually can have a noticeable effect on your emotions, on your mood, on how you behave in some really dramatic ways. So it's kind of the interface between something that seems irrelevant to your life, chemicals, and your behavior that's really relevant to your life. I generally avoid chemistry at all costs, but this is one area where it's really relevant. Okay, on the next overhead, what I'm going to do is I've broken down what goes on in this overhead into about 7 or 8 steps. And we'll go through those. I'm going to come back to this repeatedly, this one, so it's okay if you don't have it yet. The bottom left, acetylcholine, that points to the neurotransmitters, the little dots. So these are chemicals that are going to be dumped out and that can fit into the channels there. So I'll come back to that in a second. So step 1 of anything that's going to happen in the, so this is, you're going to need a long list for this, but it's nice to have it on the same page if possible, or not. The action potential comes down. This has always been stimulated by some other cell or something in the environment. And what it does when the action potential comes down, all that sodium rushing in, potassium rushing out, when it gets there, it causes these calcium channels to open. Let me show you what they look like. Those are right here, kind of on either side of the shower head. There are more of them up here as well. But so you've got your sodium rushing in, potassium rushing out. But that change in the electrical charge causes these little kind of airlock gates to open up on either side. The calcium rushing in is a signal, and it's not very well understood, but it's a signal that causes all these vesicles. It tells them, move to the front and start dumping out your contents. So at this point, it's all more or less an electrical thing. The action potential comes down, the calcium channels open, the calcium influx causes the vesicles to fuse with the membrane. It varies from neuron to neuron, but on average, you can imagine about 200 of these vesicles are present in the synapse here, the terminal button of any neuron. As soon as they fuse, they dump the neurotransmitter into the synapse. To be precise, the entire thing, the shower head, the space between, and the next cell, that's the synapse. The space between, technically, is called the synaptic cleft. So the neurotransmitter gets dumped into the synaptic cleft. People tend to refer to the space itself as the synapse. It's not exactly right. So now you've got this neurotransmitter, and this can vary. There are at least 20 different neurotransmitters in humans. Gets dumped into the synapse. I abbreviate it NT. It then diffuses across the synapse, so it's just floating. But the space is really, really small. You can see here in the picture up here, you get a sense this is a muscle cell right here, but relative to the size of the terminal button, relative to the size of the vesicles, the neurotransmitter doesn't have to go that far. After it diffuses across, it binds to receptors on the next cell. As I said, these could either be muscle cells or another neuron. Go back to the previous one. So we've got the signal comes down, calcium rushes in, vesicles fuse, neurotransmitter gets dumped, it floats around, it binds. This is the best I can do for a drawing of a receptor. I've got a couple of them here. A receptor, frequently they are two parts, and it attaches this chemical. There's a little site. It's like a lock and key, and the key goes into the lock, and it causes a slight change in the shape of the receptor there. And when it causes the change in the shape, it creates just a small enough crevice so that sodium can rush in. Sodium is a very small molecule, so all you have to do is tweak it just a tiny bit, and all of a sudden the sodium can rush in. And we know that if sodium rushes in, it causes an action potential. It also can cause a firing of a muscle cell here. The more receptors that you have, the more likely you're going to get enough sodium rushing in to cause this next cell to fire. The number of receptors here is going to be influenced by a lot of things, but you might just have a gene that causes you to have more receptors than someone else, in which case you're going to be more likely to have signals passed on. We'll see what the implications of that are. So it binds to the receptors. This causes sodium to rush in. Depolarization is just the word that when you get a quick change in the electrical charge of this next cell. And so if you've got, once that occurs, then you have the neurotransmitter. I have the hardest time with this word. What's the past tense of reuptake? Reuptook and reuptaked, taken up again. Whatever, it gets taken back up into the cell that originally released it. It gets repackaged as vesicles, and it can be used again. What I didn't write there, and I probably should write, there's sort of a little race that goes on in the synaptic cleft that, I'll come back to this in a second, that you have, it comes off of the receptor here, and it floats around, and it might land in another receptor, but eventually you've got these little molecules here that take it back up and repackage it. However, they're also, I've written down in purple here, acetylcholine esterase. So I said that acetylcholine was the neurotransmitter here. Acetylcholine, so then this would be called an acetylcholine reuptake site. But acetylcholine esterase would be a chemical, an enzyme that's in the little space between, and it's like a little Pac-Man that eats up the molecules of neurotransmitter to get them out of there. Your body wants to clean it out as quickly as possible. So this neurotransmitter has a couple of eventual fates. It either gets taken back up or it gets broken down into its component parts. But either way, your body clears out that synaptic cleft. So there was the last one. So neurotransmitter reuptake and enzymatic breakdown. And so once you know what's going on there, it allows us to talk about how your body can use these synapses to influence your behavior. That seems like a lot of details to go into for one little thing, except that all of the neurons in your body work the same way. All of the neurons in every mammal, in every animal, work the same way. Signal comes down, calcium rushes in, neurotransmitter gets released, either stimulates or doesn't stimulate the next cell, gets taken back up or broken down, the whole thing starts over again. It's a process that is really consistent across all different groups of organisms. All right, so here's our first take-home message. We've got 4 of them today. But it's at the synapse where the action potential gets converted from an electrical signal to a chemical signal, releasing neurotransmitters, stimulate tissue or another neuron. It seems odd if there's another neuron next to it, and so we had some stimulation, and the signal moves down one neuron, now it gets to another neuron, and it has to jump across in order to stimulate the next cell. That seems like bad design, doesn't it? Why not just have the neuron go all the way from the place where you detect the sensation to where you want to have the activity, the muscle or something like that? Any ideas why it would make sense? Like with your toaster, you don't plug it into the wall, have the cord go halfway, and then lay another wire right next to it and hope that the electrical signal jumps across, because sometimes it might not, right? So think about why it might be that we are built in such a way that there has to be this jump of the signal at some point, that it can't travel perfectly. Let me give you one possibility. If you listen really closely, do you hear a high-pitched whine? Yeah. I think it's the fan on this overhead. It's really annoying. If you think about the seats in this room, I've sat in this room. I took Astronomy 3 here. I think they're the same covers. They're really scratchy on the backs of your legs if you focus on it. If you think for a while your shoes might be too tight, especially after you sit for a while because the blood pools in your feet, it can't go all the way up, and so your feet get bigger and bigger over the course of lecture, and they start to hurt. These lights, these fluorescent lights, actually have a flicker to them when you're not looking at them. Your peripheral vision can detect them. If you focus on them, there are a whole bunch of things in the world that are actual external signals that are coming into your brain. Do you want to be aware of those? Ooh, it's going on. No, you don't want to be aware of it. You want to be able to focus on it if you need to, but you want it to just dissipate before it gets to your brain. And so having your body built in such a way that the signals have to jump across from one neuron to the next gives you a way of filtering out all the signals that are coming in. And you can get them if you want, but they can go away so that you can focus. And that's an important thing. Now that we're all focused on all the horrible things that are going on in this room. All right, let me tell you about part of the brain where neurotransmitter function is pretty interesting. There was a guy in 1954 named James Olds, and I think I mentioned him earlier because when I was talking about scientific method, he was doing something that was not very scientific. He was taking rats, they've done it with mice as well, and sticking an electrode into the brain. And once it was in the brain, he'd stimulate cells. And then he'd see, what does the animal do? So there's not a lot of scientific method going on here because he wasn't testing a hypothesis. But he wanted to see, if I stimulate certain parts, does anything happen? So mostly, he'd stimulate it, and he couldn't tell what was going on because the animal couldn't tell him. But they sort of just sat there like, all right, whatever. But he found one spot where if he stimulated it, he felt like it made the animal really happy. And it's dangerous as a scientist when you start describing your animals as happy. But he said that was his perception. It looked like the animal liked it, and if he did it again, they were even happier. So he rigged up this apparatus where he could implant the electrode right into the brain, and then he could stimulate that part of the brain whenever he wanted to. Now, we've also talked about the fact that you can build cages with levers. Remember I talked about the cages where you push the lever and the food comes out, and they learned relatively easily how to do it. So he had this idea. He said, well, I'm going to see if they really do like having this lever, this part of the brain stimulated. And his idea was to use revealed preference. He said, I'm going to hook up the electrode to their button in their cage so that revealed preference, meaning I can watch. If they want to stimulate their brain right there, they will. So he trained them so if they push the button, they get a little zap in this part of the brain. And he had moved it all around, and so he had it hooked up. Sometimes it was one part of the brain, and they wouldn't push the lever at all. This part of the brain, after he hooked it up, they would sit there pushing the lever about 100 times per minute. So more than once per second. And they were getting stimulated by it. He then set up their cage so they had one lever that was for food and one lever that was to get stimulation in this part of the brain. And he watched, and they'd push the lever for the stimulation over and over and over again. This is why he called it the do it again center in the brain. Oops, sorry, I should let you write down. Electrode stimulation in certain brain areas of the rats. He found that the rats would do anything to get stimulation there. The problem was he went away, he went out to lunch that day, came back an hour later. They were sitting there still pushing the lever as fast as they could. Went home, came back the next morning. They were sitting there pushing the lever as much as they could. And after about 2 days, they die. They die of starvation. How crazy is that? They have one lever right here. All they have to do is, every 6 hours push the other lever just once to get a pellet of food so they can go on. They won't do it. They just keep pushing the other lever until they die of starvation. So his name, the Do It Again Center, is pretty apt. He found that he could get them to learn really complex mazes if he put the lever at the end of a maze. They would get faster and faster because they wanted to run there and they wanted to start pushing the lever. So the obvious question is this. Why would evolution have built us to have neurons that make us feel really good when they're stimulated? Like, why was the rat built this way? I'm going to have you think about this and talk about it with someone else. Don't answer. Similarly, I want you to think about this. What behaviors are tied to human pleasure centers? We have the same places in our brain. There are about 2 or 3 or 4 of them. People disagree, but a small number. And if you get stimulated there, it causes you to want to do whatever gave you that stimulation previously. It causes you to want to do it again. I'm trying to think of my own experience with this. I don't remember if I told you this or not. When Mean Genes first came out, it's very bizarre. You spend all this time writing a book, and I was really excited, and Terry was excited about it. And then the book comes out, and it's in bookstores, and you have no idea if people like it or don't like it, if it's selling copies or not selling copies. And it's the most anticlimactic thing. You know, you've had this big book release party, and then it's like, oh, I don't know if it's doing well or not. You know, my parents said they liked it, and they bought like 20 copies. But, you know, on Amazon.com, have you ever looked? They have the sales rank, Amazon sales rank for books. How many of you have seen this? So some of you have said, they rank every single book there. And so you can look, you know, there's like Harry Potter, you know, number whatever, one, right on down to, I give my friend Michael Cooperson a hard time because he wrote a book about Arabic biography and the Middle Ages, and his book is ranked about 2,145,000 something. Not a very high rank. But here's what I discovered is that every hour at quarter after the hour, they update their rankings. And so I'd sit at my desk, and I'd wait for the clock to get there, and then I'm hitting refresh, 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 because I wanted to know what the rank of Mean Genes was. All it was for Amazon, and it wasn't like I was going to get, you know, sent money based on what that was. It has nothing to do with anything. But all I wanted was for that to, the new ranking to come out. I wanted it to be a better number. And so it made me think about these rats, because I'd be in my office, and it might be, you know, midnight or something like that. I'd be like, I have to stay till 12:50. I have to just see one more click to see what it was. And, you know, I'd keep track of, you know, what the ranking was. And I had some satisfaction because Terry was in Boston doing the same thing, sitting there. And, you know, it was all we could do. I stopped eating for, you know, long periods of time. It even got worse because we came out with a version in Germany not long afterwards. And there's an Amazon.co.de for Germany, and they updated theirs on the half hour, and so all of a sudden I could go to that one. I could look at what the Mean Genes Germany ranking was, and then one came out in Japan, and they have a site, and I couldn't read any of the Japanese site, but I could see the number, and I would plot that. But I got some sense of what a do-it-again center is like. But in general, which behaviors do you think are going to be tied to human pleasure centers? Is it going to be things like solving math problems? Is that going to make you want to do it again, do it again, do it again? Not generally. What do you think is going to be important in terms of the design of our brain? Exactly. Yes. So things like achieving some higher status. That's how, in my mind, I equated being so excited about the Mean Genes thing. I thought that if I just sold more copies, people would like me more. Turned out not to be true. But what else? So you get a mate, or you find shelter, or you get food. Food is a big one for stimulating that part of the brain. All of these behaviors that are linked to reproductive success. So here's our second take-home message. That our brains are built with pleasure centers, and stimulating them is so pleasurable that we want to behave in ways that repeat this. So your brain has set up a reward center. Now, as long as it links in behaviors like finding food, getting shelter, getting a mate, achieving status, staying safe, all of those important things make you feel so good that you want to do it again. It's a good way to build an animal. I just put this up because the best maze runners were rats that had been, and mice here, that had been trained through the use of these do-it-again centers. So it's a very powerful tool that your brain has for eliciting certain behaviors. Is so pleasurable. Here's a brain just with certain parts of it lit up, primarily right here in the center. It's an area called the limbic system. The lighter the color, the more intense the color is, the more activity that's there. That is the brain of someone where the pleasure center is being stimulated. Things related to sex should give us lots and lots of stimulation of the do-it-again centers. Not surprisingly, given that it's fairly closely tied to reproductive success, right? All right, well, let's look at what's going on in these pleasure centers. I'm going to put the same drawing. You don't have to do it again because you already know it. But this is a specific case where at the pleasure centers, the neuron gets stimulated in response to some behavior that you've done. But there is a neurotransmitter called dopamine. As I said, there are lots of different neurotransmitters. But in the parts of the brain where the neurotransmitter in the vesicles is dopamine, we're going to see it associated with a lot of pleasure. So there are adjacent neurons here that have dopamine receptors. There's going to be an enzyme in the middle there that is a dopamine breakdown enzyme. I don't know specifically which one it is, but there's something there that breaks it down. I'll move this up. The receptors on this adjacent cell bind to dopamine. They cause that cell to fire. Happiness ensues. So the places in your brain where you've got dopamine being released and you have cells responding to it because they have lots of dopamine receptors that are really efficient, they cause you to feel good. When that researcher, James Olds, was sticking the electrode into the brain and stimulating it, essentially he was hitting the postsynaptic membrane. So he was getting it in here, and he was giving a little electrical shock right here, which started an action potential. So it started an action potential. This cell here doesn't know any better. It thinks, oh, a bunch of dopamine just got released. That's why I'm firing. That's where the happiness comes from. And so all that matters is that this cell with the dopamine receptors fires. So let me give you an aside that is very relevant here. Same thing, same diagram. How many of you have done cocaine before? Wow, you guys are really clean. Dopamine, if you consume it, somehow inject it or somehow get into your system, snort it, comes right here into the synaptic cleft. And these are the dopamine reuptake transporters. So they bind to the, normally the dopamine gets released, binds here, then it gets taken back up here, recycled. Cocaine fits into the reuptake transporter and just sits there. So what do you think is going to happen? You take a bunch of cocaine, it blocks reuptake of dopamine. So what happens to the dopamine that has been released into the synapse?
It all has to get used because it can't be reuptaken.
Yeah, so it can't get reuptaken, and so it sits in the synapse. It bumps into the receptor again, and it says fire again. And so you've had one stimulation on the presynaptic cell, and from that it then says fire, and then it says fire again, and then it says fire again. And so this cell that its firings you interpret as happiness, this cell right here is like, woohoo, it's very excited because cocaine binds to the reuptake receptors, blocks them. Dopamine remains in the synapse, and so the pleasure gets intensified. Importantly, this is what's going on. There is a message that your brain interprets as something good is going on. You just got shelter, got some food, increased your status, had reproductive success. Your brain thinks that something like that is going on, and we have evolved in such a way that whenever you get that feeling, whatever you did to get it, you want to do it again. This is why cocaine is problematic, because you didn't actually get any food. You didn't actually get a mate. You didn't get increased status. All you got was the feeling that you're supposed to get when you get it. When they put rats into cages where they can push a lever and they have cocaine injected into their bloodstream, they behave exactly as if they had the do-it-again center being stimulated. So same thing. You can put a lever for cocaine, a lever for food. They push the cocaine lever over and over and over and over again until they die from starvation. So certain things are more effective at producing pleasure than the ones that we were built to behave properly. So this is a problem. It explains why it feels good, but it also explains why it's so hard, once you've done it, to not want to do it again. It's stimulating the part of the brain that is built to tell you when you've done the right thing. So it's troubling. The dopamine receptor doesn't just, it functions sort of as an all-purpose happiness kind of neurotransmitter. It's tied to lots of behaviors. Yeah, question?
Why is it that if you go through cocaine, like, more you do it, the shorter your high is?
The question is, why is it that if you do a lot of cocaine, the more you do it, the shorter your high is? Your body responds. Remember I said initially that you have genes that code for how many receptors there? If there's lots and lots of dopamine there, your body thinks, whoa, we must have made too many receptors. We've messed up. We have to have it calibrated so the right amount of the behavior gives the right amount of happiness. And so the cocaine causes more dopamine stimulation, which causes your body to produce fewer of these receptors, so it becomes slightly harder to get the response. Now, the dopamine part of the brain, those pleasure centers, are also associated with novelty-seeking, risk-taking behavior. Those also give you a bit of stimulus. This gene that I was just talking about that codes for the dopamine receptor, humans are polymorphic for it. Remember we had the word polymorphic earlier in the term, meaning multiple morphs, multiple different shapes. And in humans, there are two versions of the gene. We just call them a long version and a short version. They produce slightly different receptors for dopamine. It's still like an airlock gate that when the dopamine lands, it causes it to open up, which then causes sodium to rush in. But one case, the long version of the gene is more responsive. In other words, it binds more easily to dopamine and it opens up more dramatically. And then there's a short version of the gene that's a less responsive receptor. And their frequency in the population, about 20% of the world carries at least one copy of the long receptor. These people get more pleasure from the release of dopamine than other people. So a standard behavior that releases dopamine, if you've got the long version of the gene, you're more likely to feel really good about whatever it is that you just did than the person that has the short version. From a paper that was published relatively recently, here are some things that are associated with the different carrying the short gene or the long gene. They have a ranking of novelty seeking, and you can think about this in your own life. How novelty seeking are you relative to some of your friends? Some people are more or less. Question?
Addictive behaviors, that tend to become the drugs or
Uh-huh.
are they more likely to have the long version?
Yes, great question. The question was people who have addictive personalities, what does that mean? Is that related to, do they carry the long version of the gene? And the answer is yes. Question?
So they have the exact opposite effect, or is it related in any way in terms of like how bad you may feel about something?
It's a good question. The question is, does it relate to how bad you might feel about something, that you got too much pleasure than you should have? I don't know the answer to that. I'm not sure. If there are guilt centers of the brain as well that are related, it wouldn't surprise me. But here's the difference that they found, that depending, remember, we carry 2 copies of every gene, so you can have 2 copies of the short, one, long, or 2 long. And the more long genes that you carry for this, the more responsive your cells are to dopamine. Individuals who tend to have more of the long gene, or at least one copy, are impulsive as opposed to reflective. These are based on actual analyses of behavior, but also analyses of people's responses on questionnaires. They tend to be more exploratory. This is true in animals as well, versus rigid. They tend to be fickle. They'll change their mind a lot as opposed to loyal. They tend to be really excitable as opposed to stoic, rigid. The people with the short versions, more of an even keel. They tend to be slow-tempered versus quick-tempered. These tend to be frugal versus extravagant. No difference as you get older or younger. No difference between males and females. I'll tell you something interesting about this. One of the most interesting jobs that I have is I talk to the FBI. They have what's called a behavioral analysis unit. And don't tell anyone I told you this, but their behavioral analysis unit, one of the things that they do is they want to find people from other countries, say somebody who works in the embassy for a different country that we view as having information that we might want, and they need to find people that they say, hey, we'll give you some money or we'll help you out, we'll do something nice for you if you tell us some secrets. The problem is if you're a citizen of Russia or China, and you tell the government in the U.S. some secrets that you have, and they find out back in your home country, that's bad news for you and your family. So the act of telling a secret, essentially committing treason, is a very, very risky behavior. So the FBI is interested in finding out, are there ways that we can identify risk takers? Because out of the hundreds of possible people, if we could very quickly focus on the few people who are more likely to say yes than no, we could save ourselves a lot of time and money. So what they've asked me to do is come in and tell them how, just by looking at a group of people, I can say, that gal, that gal, that gal, and that guy are probably who you should focus on. So there's a lot of interest in figuring out, all right, how can we figure out what do they have? Obviously, if you could get their DNA and analyze whether or not they carry the long gene, that would be good. Tough to do that, though. So you want to look for behaviors that are associated with it. Are they exploratory rather than rigid? Are they excitable? Are they quick-tempered? One of the most bizarre things that turns out to be correlated with this, people who have one or more copy of the long gene tend to like spicier foods more. Very, very strong finding research-wise where they've looked at the genetics. And so think about this in your own life. I actually, when I went and talked to the FBI once, I'm at their headquarters in Quantico, and I said, I want to do this with the group of agents. Before I tell them anything, I want to ask them all to fill out a questionnaire about who likes spicy foods. And they told me I couldn't do it. Why would they say that? Because they said that the group of agents that I was working with are also approached by people in other countries who want them to tell secrets about the U.S. And if I found out who likes spicy food more than the other ones, I'd be essentially calling attention to the people who are at most risk of turning on the U.S. And so they said, it's not funny. We don't want to play like that. But they do want to now look into people from other countries and find out, all right, who is it that wants the spicier foods or not spicier foods? So it's a quick and easy way that you can get a sense. It's not completely correlated, but it's a good predictor of who's more likely to have the long gene. Now, here's another question to contemplate, also related to dopamine receptors. People have thought about this. The idea of leaving home and making some long migration is also risky. So some anthropologists had the idea, wow, we could then make predictions given that humans likely evolved in the first place somewhere either in Africa or Asia or both simultaneously, it's unclear. But if that's where we started, the further away you get from there, you're talking about people who have left home and gone far distances. The ones who make it are likely to be big risk takers. And here's what they found. This is a, I think, stunning graph. Before I put it up there, who do you think are going to be the parts of the world where the percentage of people carrying the long gene for dopamine receptor is at the highest percentage? Antarctica? Yeah, nobody living there. Where? South America, yes. And who should be right behind South America? North America. Yes, and you can look at maps of dispersal over evolutionary time and make predictions. And here's what they found. Migration distance from, and people all disagree on where they should say the original point of evolution is, but they found that the further the distance was the higher the proportion of individuals with the risk-taking gene. In particular, among Chinese and Japanese, 5%. These are people living in those geographic regions. Africans were at about 16%, have at least one copy of the long allele. Europeans are at 20%, Pacific Islanders 21%, North Americans 32%, Central Americans 42%, South Americans 69%. When you get a result like that, that's stunning to have made that prediction and have it come out so strongly related. So it may cause this responsiveness of your dopamine receptor may be responsible for a lot of different behaviors that people have. Whoops, sorry. Yeah, so there's a lot of debate. A lot of people say, well, this is weird because we would expect Africans to be lower than Asians. Unclear there. So, these are relatively recent results. People are trying to figure them out. Sorry, I had all these pictures I was going to show of cocaine, but none of you have ever done it, so we don't have to worry about that. Risk takers. So here we've got a guy going off a cliff here. I would bet money that this guy's got 2 copies of the long allele. Same thing with these guys. And so people have started now looking into different behaviors and then measuring, looking at which copies they carry. People who are explorers, here we have in the Himalayas, or doing crazy stuff. Anyway, so this will be our third take-home message, that risk-taking stimulates the brain's pleasure centers. And some brains, by that I mean some individuals, are going to experience more pleasure than others for a given activity. It seems like a fun thing when you talk about it being risk-taking and novelty-seeking, but as the question up front here earlier was, what about addictive personalities? Same thing. So people's likelihood of smoking, of doing drugs, are also significantly increased if you carry the long receptors. So there are certainly trade-offs here. In the questionnaire, some of the questions that they ask to figure out what type of personality you have, and I remember, as I've told you, questionnaire data are notoriously inaccurate. That's why you got to look at the behaviors. But they have things like, I have sometimes done things just for kicks or thrills. So people have to put yes or no for that. Think about what you would put. Or I think things through before coming to a decision. Question?
Which genes carried more often, did you say, worldwide, 20%?
So if you average everybody, the short version is carried more commonly. So the big risk-taking one is at a lower frequency. You get one from your mom, one from your dad. So you could get 2 short ones, a short and a long, or 2 longs.
It's not like a dominant thing.
No, it's the more long receptors you have, the more stimulation from dopamine you get. Because if you got a short version and a long version, on the neurons in your do-it-again centers, you have different kinds of receptors. Both genes produce receptors, so you have some that are really responsive and some that are not responsive. But the more you have that are responsive, the more likely a little bit of stimulation causes the next cell to fire. I was so happy when I first learned all this, just because it helped me understand things so well. Terry, my co-author on Mean Genes, jokes all the time that anywhere we go out, all I want is for them to have spicy peppers, and I will sit there at dinners just eating the spicy peppers, he says, because I'm so bored with life that I need some kind of stimulation. I don't know what it was. Ever since I was a little kid, all I wanted to do was eat the spicy peppers until I got old enough to ride a motorcycle. But knowing it now, I realize that it's something you have to be aware of. So let's look at another neurotransmitter, something called serotonin. Here we have more crazy risk takers. Oh, here are our peppers. Just seeing them makes me happy. Serotonin is another neurotransmitter. We write about it in Mean Genes as another happiness neurotransmitter. It affects appetite, sleep, anxiety, mood. This is one of the difficulties of linking up the genetics of what receptors you carry with the behaviors you exhibit, because you've only got 3 or 4 do-it-again centers, but there are all sorts of behaviors that your body wants to encourage. So there is lots and lots of overlap. Certain parts of the brain fire in response to multiple different types of stimuli. So it's not really clean, like, oh, when this part fires, then this behavior ensues. So think about this. I'm saying that serotonin is a neurotransmitter. When I say it's usually inhibitory, what I mean is that certain neurotransmitters, when they bind to the receptor site, they cause the next cell to be less likely to fire. So they clamp down, they close up the sodium channels. But that's irrelevant to us. It's just has some effect on the next cell. So after the serotonin is released, it has its effect on the other cell. It gets reuptaken by the previous cells. What if you block reuptake, the way dopamine reuptake could get blocked by cocaine? What if we blocked the reuptake of this one? Normally, serotonin makes you content and satiated. If I block the reuptake of it, what's going to be the outcome? Now think about it. Serotonin gets released. When serotonin is released, release of it causes you to be content and satiated. So you're super content and satiated because it stays in the synaptic cleft and it keeps attaching itself to those receptors. Is that going to be good or bad? Would you like to be content as opposed to discontent or malcontented? Would you like to be satiated or do you want to be hungry all the time? It's pretty good. It's, well, you wouldn't. Yeah, you maybe wouldn't want to do it permanently. So that's a good point. Now you're content for no reason. Somehow we've blocked reuptake and we've separated out the emotion that you were built to feel from the behaviors that get it. So we know that with the dopamine thing, I said, oh, block reuptake and you feel lots of happiness. But my point wasn't we should all be doing more cocaine because we'd feel so happy. In the short run, I would say that, like if we're going to get hit by a bomb in 3 minutes. But in the long run, such as 4 hours or 40 years, it's bad. In this case, you get no more depression. So depression can be treated by blocking reuptake of serotonin. So if you know anyone who's taken Prozac or Zoloft or Paxil or Luvox or Celexa, the fact that there are so many of these should be a tip-off that lots of people are selling them because it's a desired effect. You've probably heard of them, and I didn't give them the name, but they're SSRIs. Have you heard of those letters before? Stands for Selective Serotonin Reuptake Inhibitors. So we inhibit reuptake of serotonin. There is one dopamine reuptake blocker, Wellbutrin, that's also used for depression. That should make you think, well, wait a minute, cocaine is a dopamine reuptake blocker as well. Should they be prescribing that? And, it depends on how quantitatively, how much do you control it? And cocaine is out of control, whereas Wellbutrin can control it at exactly the levels of stimulation that lead to depression going away. So this can be very useful if, for some reason, you've been built in such a way that your receptors don't respond well to serotonin, or you don't have that many, or you don't produce enough serotonin. All of those things can lead to you to being slightly more depressed than someone else. And by taking these, it's a way to artificially increase the effect of serotonin, and you can have a good outcome from it. Here's something I want you to think about. Given that it also tends to make people feel satiated on average, I want you to think about 4 different ways that you could design a diet drug based on this knowledge. And by this I mean, okay, we know serotonin makes you feel satiated, so to get that feeling of satiation, one way is to have people eat a lot of food. That's not going to help as a diet drug. Alternatively, we can have them feel as if they've eaten a lot. So if you keep serotonin in the synaptic cleft, you can get people to feel satiated even when they're not eating food. So how could you do that? You could block the enzymatic breakdown of it, right? So it sits there longer. That would be one way. You could block its reuptake. That would be another way. I've already given you 2. So I want you to come up with more than that. I want you to come up with 5. But you see what I mean? You have to come up with ways that you can increase the effectiveness of the serotonin that is in the synaptic cleft. Look at this whole field of chili peppers. So this is something we don't have time to talk about today, but I said, hey, it would be a nice way if you want to figure out who could be a spy, find out which gene they carry. There's more money to be made in this, and that is this. Find out who carries the long gene and know that they are more likely to become addicted to drugs. If I'm running an insurance company, what could I do with this information? Find out who carries the long version. Don't insure them. Right? Well, I mean, if I'm a publicly held company, my job is to maximize shareholder benefit. So by excluding all the people who are likely to be risk takers or drug addicts eventually, even if it's only 1/100th of 1%, I can increase the amount of money because we won't pay out as much, which is great, right? It's very scary. It is great from an insurance company perspective. However, if they use this information to discriminate, this can lead to disastrous consequences that all of the people who, through luck of the draw, got the wrong gene for their dopamine receptor suddenly can't get insured, and that can lead to very bad outcomes. Currently, 37 states in the U.S. allow insurance companies to find out what genes you have and to modify their coverage or how much they charge you based on it. So it's the kind of thing where you want to find out people that you're voting for, where do they stand on insurance companies using genetic data, because it can lead to you suddenly not having any insurance because you carry the wrong gene. Okay, here's a... What's that? Can they force you to find out? No, they can't force you to find out. However, if you're sitting in a chair like this, after you leave, if there's a hair of yours that happens to be on the back of this, I can take that hair. It's very tough to get DNA from hair, but it can be done if you leave enough. If I kept coming here. And so they invite you in for an interview for one reason or another, and you've left that there, they have the right to use that. So they don't have to force you to find out, but they can find out. Okay, here we have some drugs, some cocaine, some ecstasy. How many of you have done LSD before? None of you. LSD, it stands for, it's also called acid. It's lysergic something, dimethylamine acid, something lysergic acid. It is a serotonin mimic. When you consume it, it gets into the synaptic cleft, and it acts just like serotonin, meaning that it binds to the receptor that normally only binds to serotonin. And so you've had no release of serotonin, but these other cells suddenly are feeling content, satiated, but in extreme amounts. And it causes people to feel really happy, but it also stimulates a bunch of other cells. It gives them hallucinations and all sorts of other stuff. How about ecstasy? How many of you have done ecstasy before? I love that you... No one. How many of you have a friend who's done ecstasy? Yeah, I know that guy. Yeah, he does it. Ecstasy is a tricky one because I, somewhat bizarrely, have never done any illegal drugs in my life. But the closest I ever came was with ecstasy. I knew a guy when I was in graduate school, and he was really into it, and he worked in a lab where he could make it. And so for me, I'm like, wow, well, at least I know that it's going to be what it's supposed to be. So I did all this research on it, and it sounded amazing. There were these therapists who were writing saying that I can, make more progress in one session with a couple that's both taking ecstasy than I could in a year of therapy, that they had insights into their relationship, into themselves, and they persist after the drug wears off. So I thought, that's pretty good. And so there were a lot of people saying good things about it, but there were also people saying, we just don't know enough about it, we don't know how long it leaves its marks on the brain, and so on. And so for being someone who tends to be a risk taker, I decided that it wasn't worth it. I had these weird ideas that I wanted to be on the Supreme Court someday, and I always thought, I just want to be able to say, no, I haven't done any illegal drugs. Anyway, since then, here's what they've discovered. There are two effects that ecstasy has. There are a bunch of different pictures of what it can look like. Ecstasy increases serotonin production. So it causes the vesicles to be packed with more serotonin. So one release causes a lot more serotonin in the synaptic cleft. It also blocks reuptake. So you get a double whammy. You release more of it. You don't let it get taken back up into the other cell. Huge amounts of serotonin effects, happiness from the release of it. However, let's see if I wrote it down here. There is a downside. In one study, and this is a very poorly done study because the sample sizes are so small, nonetheless, it's the best I can give you. There was a study where a bunch of monkeys were given ecstasy twice a day for 4 days. So not a super huge amount. Think of it as a terrible spring break binge. And they did this ecstasy time twice a day for 4 days, never did it again. They came back and they looked at the serotonin-producing cells. 7 years later, they were still damaged. Here's what it does. It damages, and they say permanently, this was 7 years, it's unclear, cells in two parts of your brain. The cerebral cortex, parts that are used for learning, and the hippocampus, which is important for memory. In other words, it causes these cells to run just on overdrive for brief periods of time, but it wipes them out, even in a short period of time. And so if it's important to you to have sort of maximal abilities of learning and memory, as it is for me, it causes, at least for me, it causes me to think, I don't know that that's worth the trade-off. I know a lot of people have done it, and they say, hey, it's really great. And I think, I have a career where it's important that I can be as smart as possible and remember things as well as possible. And so the risk is a little bit too high. I'm barely hanging on as it is. So I'm going to move from this to a more general question of how do drugs work. And in almost all cases, here's how it goes. They interfere with neurotransmitters. And they do it by either blocking the release of the neurotransmitter. So there are lots of ways you can interfere. For instance, you could block the calcium channels of certain cells so that when the action potential comes down, calcium doesn't rush in. That would be one way to do it. You can over-facilitate neurotransmitter release. So you could cause maybe the calcium channels to be more responsive in those parts of the brain, the do-it-again centers, or the dopamine release centers or the serotonin release centers. Or you could figure out ways to increase or decrease the breakdown of the neurotransmitter in the synaptic cleft. So as I said, you can, if you inhibit the enzyme, the neurotransmitter will stay there for a longer period of time. What else could you do? You could block or facilitate binding to the receptors on the other side. These are not the only ways, but you see what I mean? Where because all the parts of the brain in all the different species of organisms in the world, of animals, work in the same way, you can come up with generalized ways of making drugs just by messing with what goes on in the synapse. So I've talked about all these illegal drugs. How many of you have done caffeine before? Doesn't it feel good to finally tell the truth? I asked one of my, I'm not accusing you. Yes, I was accusing you of lying. Here's why. I once made a deal with an LS15 class. I had them all close their eyes, and I said, in order to get them to tell me the truth, I said if they told me the truth on this one question, I would let them choose one topic on the final not to cover. And so I had them all close their eyes, and I said, raise your hand if you've ever done ecstasy. And they were actually honest, and guess what percentage of them had raised their hand? 75%. For the first time in my life, I felt really out of touch with the world. I had no idea. I was stunned. Anyway, that was a few years ago. I think things have maybe changed a little. All right, so I want to talk about one drug that is legal, caffeine. If you look around, you can find animals yawning everywhere. Yeah, if you say they're good teeth, it's because that's in a zoo. Anytime you have good teeth and fat animal, let's see. Oh, sorry, we'll go back. We'll leave this one. What's that? Do they put braces on animals? Not that I'm aware of. There's a chemical called adenosine. It is a byproduct of cellular metabolism. In other words, the harder your cells are working in your brain, these neurons, the energy source they use is something called adenosine triphosphate, ATP. You don't need to know that, but you do need to know this, that the more your cells are active, the more of this adenosine they produce. Over the course of the day, adenosine builds up. And here again, our shower head model. It turns out that as they release adenosine comes out of these neurons, in addition maybe to dopamine receptors, serotonin receptors, acetylcholine receptors, they also have adenosine receptors. And when adenosine lands in this molecule, in the receptor right there, it sits there and it causes the ion channels, the sodium channels, to be less likely to open up. So what does that mean? The sodium channels are less likely to open up. The next cell doesn't want to fire. And if the next cell doesn't want to fire, what do you experience that as? I think about it when I sit in my office and I'll be writing this textbook now, and I have deadlines every 3 weeks. I have a big deadline, and almost without fail, I spend 2 or 3 nights in my office right before the deadline. And I'll be there, and it'll be 4 in the morning. You've probably had this experience where I'm sitting there, and I'm telling my brain, come on, focus, just think about it. You know, and sometimes I'll sit there, and I'll start hitting my hand or something. Just all I want is for my brain to stay alive so I can finish the task I have. And the cells in my brain, they're like this. No. And they sit there. And why is that? It's because all the adenosine that I've been producing is filling up these receptor sites, and it's telling them, don't fire. Forget about how much stimulation you get from the other neurons. Don't fire. So it's like a brake on neuron activity. Then when you go to sleep, the adenosine comes off the receptor, it gets taken back up into the other cell, and it gets recycled. Question? Why does it happen during sleep? Sadly, the answer is I don't know. Sleep, in this textbook I'm writing, I wanted to actually have one of the chapters be on sleep because I'm really interested in sleep. I love sleep. And I had to cut it out because nobody knows anything about sleep. So we're in the dark ages, literally, when it comes to sleep. No one knows what goes on. We know that adenosine gets reabsorbed. That's not the only thing that keeps you tired. It's not clear whether it's light, dark cycles that causes your body to send out the messengers that pull the adenosine off. We don't know. So all I can tell you is that happens. I don't know why, though. So here's where caffeine comes in. What I have here is actually coffee beans. Here they are after they're roasted. Believe it or not, I've never had coffee in my life or tea. I don't like the smell, but I do like Diet Coke. I've had a lot of that. Why are the ion channels open?
Why would that make your cell likely to fire?
Because in order for a cell to fire, you have to have a depolarization. You have to have a change in the charge inside. And if you have them open all the time, sodium is at equal concentrations on both sides. So the cell cannot fire. Here's what caffeine does. It masquerades as adenosine. Chemically, it looks really, really similar. So that's problematic, right? That would make you think, oh, well, it masquerades as adenosine. It lands on these adenosine receptors, causes them to be less likely to fire, so it should make you tired. However, it doesn't make the neurons less likely to fire. So it blocks the place where adenosine could have its effect without changing the cell's likelihood of firing. So the more of your cells you can get filled up with adenosine, the better off. Question? That would be my advice, except that caffeine has this great feature. It's actually better than adenosine at filling in those receptor sites. It can knock it out. So it can make you untired. So you don't have to take it ahead of time to fill them up. The result is that, so I've just put that it prevents accumulating adenosine from inhibiting the brain. It also can knock it out and reduce the effects of adenosine. If this were the only reason you got tired, you could stay up forever just by using caffeine, but there are other breaks to nervous system activity. This is just one of them. Pretty great, though. One of the reasons I'm interested in caffeine, I don't consume a ton of it, but Terry Burnham consumes huge amounts of caffeine, huge amounts. When he and I first became friends, I was staying at his place overnight, and I got up in the morning, I saw him taking some white pills. I'm like, what are you doing? What are you taking? He said, oh, it's caffeine pills. I was like, wow, that's hardcore. Why? He says, because I want to be alert. I need to take some caffeine. I said, that's uncool. Sort of saying it without thinking. He said, not really. He said, think about this. Almost everyone we know gets up and they will have some form of caffeine. The difference between me and them, he said, is that I know exactly how much I've had. He said, let me show you a spreadsheet. He pulled out a spreadsheet. He had 10 years of data of exactly how much, because over time what happens is that you build up a tolerance to it. So he does what he and I call caffeine watch, where for about 2 weeks, it takes about 12 to 14 days for your receptors to go back to their normal activity, go off, and then you ease up on it again, and it has more effect, and so you would increase it, and so on. And so Terry has done a huge amount of research to find out, are there long-term negative effects of caffeine consumption? It almost certainly seems like the answer has to be yes, right? Because there's almost never a free lunch. I'm going to tell you something here that's a risky thing to say, but I'm going to tell you there is no evidence of any long-term negative consequences of caffeine consumption. None. Question in the back. Yes, good point. If you are... I would not give it, it inhibits growth in terms of bone growth, so that, in humans, we reach a final growth size, and that's that. So I'm not giving my son Jack caffeine because I want him to grow. So there are certain situations. I mean, for someone in your situation, no long-term effects. And there are some additional positive effects. Athletic endurance, here I'll go ahead, 19.5% more endurance with caffeine consumption 1 hour before a bike race. That's a significant boost. And so individuals who are training frequently use caffeine. And I had the previous one about the rat in the maze. There's increased performance in maze learning by rats that are given caffeine. If they have caffeine while they're learning the maze, doesn't even matter if they have the caffeine later when they're running the trials. It increases their ability to learn it. Oh, shoot, I forgot the overhead for the last take-home message. I'll put this, I'll bring it next time, but take-home message 4 was this: drugs can hijack pleasure pathways, tricking your brain into believing you're engaging in good and worthwhile behavior. So I have one last thing that I want to do before we go today. I told you that there's no lecture on Thursday. I'm giving a talk about engaging biology students in lecture. That sounds silly, but would you mind, and you don't have to do this if you're in a rush to get somewhere, writing down for me just one answer to each of these? You don't have to write your name on it, doesn't matter. But if you can give me just your opinion on what makes a lecture that engages you and what makes for a lecture that doesn't. It doesn't have to be articulate, but it's something I can read on the plane to give these people, when I give the talk, some actual data from you guys. It's a conference called the National Association of Biology Teachers, but here's the other one, so no lecture. And then when you leave, we have problem sets for next week, since we won't have lecture on Thursday. Here they are. Pick these up for then. And I will stop here. Thanks.